Thank you, Catalina. Good afternoon, everyone. It's my pleasure today to be presenting the research that I've been working on this summer at the Extracorporeal Life Support Laboratory. My project is 12-hour normothermic ex situ heart perfusion. One of the major problems with organ transplantation as a cure for patients with chronic heart disease is the prolonged time that patients have to spend on the waiting list. Around 350 patients a year die waiting for a heart. That's about one patient a day. Currently, there are 4,026 patients on the heart waiting list, but only 620 hearts have been donated. The waiting lists are increasing, but there hasn't been a compensatory increase in the number of donors. Currently, cold ischemic preservation is the predominant option to preserve hearts. This is storing hearts in ice on 5 degrees Celsius. When a heart is on ice, we can't assess how it would function in a transplant recipient. Also, when a heart is on ice for greater than 4 hours, there's an increase in post-transplant complications due to ongoing cellular metabolism. My project is moving us towards unlimited organ access because we're aiming to assess the functionality of hearts and include in the donor pool organs that wouldn't be considered suitable for transplantation. So the goal of this study is to use the novel system to maintain beating hearts outside of the body for a 12-hour period, facilitating organ evaluation prior to transplantation. This is a schematic I created detailing the major components and nutrient flow directions of this system. The pump under the table, here controls the flow rate and pressure, and the nutrient-rich fluid or perfusate. This comes up here, it gets oxygenated here, and this heater keeps it at normal body temperature, which is 37 degrees Celsius. It then goes to the heart via the aorta, the right atrium, and the left atrium, and leaves via the pulmonary artery and left ventricle to this reservoir, which then takes it back to the pump. Our goal is to keep the heart beating for at least 12 hours. This is a period where chronic injury to organs can be assessed and treated, and organs can also be transferred intercontinentally. Imagine being able to share hearts with countries such as France. Waiting lists around the world would be drastically reduced. So this is the timeline of our experiment detailing our research process, which ultimately leads to the heart being placed on the circuit I showed you. 4 healthy adult pigs under anesthesia were used as heart and blood donors. Lines were placed in the femoral and jugular veins to draw blood for our customized perfusate, which will nourish the heart outside of the donor. Then, while one team explanted the heart using the standard technique of cold cardioplegia, I helped another team harvest the blood from the lungs. We centrifuged the blood into its constituent parts, and then we discarded the white blood cells and platelets, and mixed the plasma and red blood cells with other nutrients to create our customized perfusate. And this is what's going to keep the pressure of the proteins in the fluid equal to 30 millimeters of mercury. Meanwhile, the other team put the heart on ice for only 30 minutes and placed cannulas in all 4 chambers. These cannulas are what we then use to attach it to the perfusion circuit, and we followed a standard perfusion protocol. We then rewarmed the heart to 37 degrees Celsius, normal body temperature, over a period of 45 minutes. And we saw a return of normal beating, as you see here. This heart is beating entirely on its own, with no electrical stimuli driving it. It is solely connected to the circuit that I showed you, and its nutrients are the perfusate I mentioned previously. Here you see a infusion cannula into the aorta, a drainage cannula coming from the left ventricle, and a pressure control cannula in the right atrium. This thick blue wire here is a temperature probe. That's what helps us make sure the heart is at 37 degrees Celsius. These red, green, and black wires, those are measuring electrical conductivity, which shows up on our EKG. So we studied several variables, including the pressures and flows in each chamber, the cardiac oxygen consumption, and markers, such as lactate and electrolytes like potassium and bicarbonate, which showed us whether or not the heart was viable, basically as healthy as it would be at physiologic levels in the human body. So I graphed the average oxygen consumption and lactate levels over a function of time. Oxygen consumption shows that the heart is alive and functioning, and we can see that this holds relatively constant over the time interval. The decrease in lactate levels that you see here suggests that the heart is engaging in aerobic metabolism, which means that the tissues are getting adequate oxygen supply. So overall, we found that the use of this novel system does facilitate the assessment of cardiac function of organs prior to transplantation. And if you can remember that we kept these organs on this pump for 12 hours, and that's more than half the time an organ can be kept on ice. So our next step would be to identify certain biomarkers that correlate with organ function, so we can determine which organs are healthy enough to transplant and which might require special treatment or observation beforehand. We also like to experiment with different types of nutrients in the perfusate that might help keep the heart more viable. So right now, I would like you to take a moment and imagine a world where there's no such thing as a waiting list. If someone needs a heart or a liver or any type of organ, one can be plucked from an organ bank and transplanted readily with minimal to zero side effects. My project is moving us towards this goal, and I hope I gave you a glimpse of this goal today. I've learned a lot by working on this project. I've learned the importance of acid-base balance, how to control and monitor electrolytes, all about the anatomy and physiology of the heart, and what it's like to work in an operating room and assist with surgeries. This project actually helped me determine that surgery is the career I wish to pursue. Before I open up to questions, I would like to take a moment to thank the people who made my time here at the University of Michigan so rewarding. I'd like to thank the ECLS Laboratory, my sponsor, Dr. Elvira Ross, for welcoming me as a research fellow and giving me the opportunity to conduct such fascinating research as an undergraduate. I'd like to thank Urop for giving me this opportunity in the first place to stand up here today and for choosing me to participate in this wonderful, amazing program. And I've enjoyed working with all of you this summer. And I wouldn't actually be here today without the encouragement of my mentor from Schoolcraft College, Dr. David O'Hagan, who first sparked my interest in research. Thank you.